All right, class. So here we're going to talk about meiosis and why do we need two different ways of separating chromosomes. If you remember back to our cell cycle lecture, we mentioned that step 2 of the cell cycle was mitosis or meiosis, but both of them we said were used to separate chromosomes, right? So why? Like why do we have to have a second way? And the answer to that is because we're going to need daughter cells with half the number of chromosomes. Okay, that's why we need a second kind of way of dividing DNA, dividing up those chromosomes, because we want daughter cells with half the number of chromosomes. All right, so the good thing with meiosis, there's kind of a good and a bad. The good thing is all the stuff we did in mitosis is still true. Um, the stages we have, the order of the stages, all that stuff is still true. We just actually do them twice. So this looks like it's a really long list and it's complicated, but it's no different. It's just the stages we did twice. And since we're doing them twice, we use Roman numbers to indicate whether we're in the first one or second one. So same stages, right? Prophase, prometaphase, metaphase, anaphase, telophase, but with ones, so we know we're in the first part of meiosis. All right, so these first 5 steps, all of that together is referred to as meiosis 1. Meiosis is actually going to have kind of 2 parts, 1 and 2, right? Makes sense. So then in between here, we are going to get cytokinesis. Okay, and then we go into meiosis 2. Same 5 steps, but now with 2. Same 5 steps, same order, just 2s instead of 1s. And then all of those 5 together, that's meiosis 2. And then there is another cytokinesis at the end of meiosis 2. All right, so, again, this list, like it looks like this gigantic list. It's long, but I mean, it's the same stuff. It's just happening twice, right? And what we've talked about, if you remember back to our mitosis lecture, what we talked about, like, here's what happens in prophase. That still happens in prophase 1, and it still happens in prophase 2. It's just happening twice instead of once when we're looking at meiosis. What's happening in metaphase? Chromosomes line up. Metaphase 1, chromosomes line up. Metaphase 2, chromosomes line up. Right? So like the same stuff is happening. It's just twice. Okay, and since we're doing this set of events, these steps twice, and cytokinesis twice, and meiosis, this is our picture. That's our first cytokinesis. Then a second cytokinesis makes 4 cells. Right, so this is meiosis 1. And then the second set of arrows is going to represent meiosis 2. Right, and the way we're going to get half our number of chromosomes with this, meiosis 1, let me tell you these. This actually, it's like, I can understand where you may think, well, you're just giving us another name for the same thing, but this can actually help. So meiosis 1 is called reduction division. And meiosis 2 is called equatorial division. Right, so we've got two divisions, meiosis 1, meiosis 2. One's reduction, one's equatorial. So one reduces, one remains equal. Right, that's what it's telling you. So if these cells, if this parent cell is 2N, meiosis 1 is our reduction division. Right, so we're going to reduce that 2N in half. So these first two daughter cells would be 1N. And then we do meiosis 2, which is telling us it's equatorial, so everything stays equal. So these last four cells would stay as 1N. All right, we don't split it in half twice. We don't want that. We just want to split it in half once. And we split it in half once during meiosis 1. That's when we're doing the splitting. And then with chromosome numbers, like we looked at with mitosis, we can do the same thing here. We just got to follow them through two divisions. Right, so if we know in this cell here that 2N equals, let's say, 20, then, these cells are 1N and these cells should be 1N, so this is 10 and 10. And then these stay as 10, right? So we can still do the same thing with the numbers that we have. You got to understand that that first division we're splitting in half instead of keeping it the same. Mitosis, from before, we always want to keep it the same. All right, and then, so I'm not getting into a lot of the specifics of how meiosis is different. There's a handful of things I could show you and talk to you about that are why is meiosis different. I've shown you one here, which is we're splitting a number of chromosomes in half. And I want to show you one other thing. In prophase 1, this thing happens. It's called crossing over. Hey, this happens in prophase 1. This is only in meiosis. We did not talk about this for mitosis. Right? And it looks like this. Okay, we have our chromosomes in our pairs. Right? So now I'm going to draw these different colors, but this is representing an actual pair. So like if this happened to be a number 3 chromosome, this is also a number 3 chromosome. So it's got to be one of those pairs. Right? So what happens here is literally what the name says. So parts of our chromatids will literally cross over each other. So you'll get things that look like this. Picture would look like this. So there's some crossover with these chromatids, right? They're physically crossed over each other. And then what happens is these chromatids will swap pieces of their DNA. So you'll get what looks like this. Right? So if they're swapping pieces of their DNA, it looks like this after we've swapped. Right? So the black chromosome has some orange on it, and the orange chromosome has some black on it. All right? These chromosomes that we've created here have crossed over. Right? And what that means is that they are genetically different. There's a genetic difference between those chromosomes compared back to our original two. So the importance of this, and like this is really, really important from biology. The importance here is that this increases genetic diversity. And that's kind of like baseline what we've got to have for things like natural selection and evolution. We've got to have a lot of genetic diversity for that to work well, right? And this crossing over stuff increases genetic differences tremendously. Now notice that like we haven't introduced anything new, right? Look at our picture. The black has orange, the orange has black, but we haven't done something like, if we were drawing this chromosome, we haven't done something like this, where we all of a sudden say, hey, look, there's a piece of DNA that's red. That's absolutely brand new different. Like we can't do that here. This crossing over process, it's like shuffling a deck of cards, right? So every time you shuffle, they're in a different order. Every time you shuffle, here's the shuffle. Every time you shuffle, they're in a different order. Okay, but when you shuffle a deck of cards, you don't just spontaneously have some brand new card that you just made in the middle of your deck, right? You have the same 52 cards. They're just in a different order. So that's what crossing over does. It only happens in meiosis. It's never going to happen in mitosis.